Hola, hola, ¿cómo están? Buena tarde. Rally, can you hear me? ¿Me puedes escuchar, Lee?
Yes.
Perfecto. Perfecto. Um, bueno, pues vamos a empezar ya pues el seminario del día de hoy. Pues buenas tardes a todas y todos. Gracias por estar aquí el día de hoy en esta primera sesión del ciclo de tópicos de fisiología y farmacología, un ciclo de seminarios que se vienen realizando semestre tras semestre. Es un gusto saludarles a quienes nos acompañan también tanto de manera presencial y también a distancia a través de Zoom y YouTube. El tema de hoy es de especial interés en medicina veterinaria, ya que platicaremos sobre aspectos relevantes de la fisiología del dolor y los circuitos cerebrales que están relacionados con la modulación del dolor, de las respuestas conductuales que se derivan de amenazas o estados de aversión. Para hablarnos de este tema, hoy nos acompaña un invitado internacional. Es para mí un gusto poder presentarles al doctor Lee Aiden. Hi, Lee. El doctor Aiden es jefe de la sección en neurociencia molecular en el National Institute of Mental Health, que es parte de los Institutos Nacionales de Salud de los Estados Unidos. Tuve la fortuna de poder conocer y trabajar junto al doctor Aiden y su equipo de colaboradores en el año 2021, perdón, y a partir de el interés en sus trabajos sobre el dolor y la modulación de este a través de neuropéptidos, eh, entre ellos el polipéptido pituitario activado de la adenylato ciclasa, el PACAP, que de hecho el día de hoy nos va a hablar sobre él. Y bueno, pues sin más preámbulo, pues solamente quiero comentar que la trayectoria del doctor Lee es por demás sobresaliente a través o a lo largo de 4 décadas de investigación sobre sustancias químicas moduladoras de la actividad del sistema nervioso y el sistema cerebral. Y bueno, pues deseando que esta plática sea muy provechosa y queden fascinados con los neuropéptidos. Um, Let me introduce Dr. Lee Aiden. Lee, I give you the floor.
Diego, gracias por esas palabras tan amables. Me hubiera encantado poder conocerte en persona en la UNAM. Pero fate has left me here in Bethesda, so I join you remotely, but feeling very much with you in person. And I thank you for giving me the power to admit people remotely. So if you hear a chime occasionally, it's just me letting people into the room. So what I'd like to do today is to highlight two brain circuits of great physiological importance for responding to pain, stress, and other threats, and how classical transmitters like glutamate function along with neuropeptide neurotransmitters to allow information to be passed across synapses in these circuits. Physiologically, especially at the molecular level, leads naturally to pharmacology, the greatest and oldest translational medical discipline. So it's especially satisfying for me that this talk is a topic in physiology and pharmacology, and that you all are an audience that includes students of veterinary medicine, which is the backbone of animal experimentation that underlies both basic and clinical brain research in these weeks before Brain Awareness Week. So with that introduction, may I embark on a tale of two types of transmitters in describing some brain circuits for pain and threat responding. I want to start with a historical perspective on the neuropeptide PACAP, which is our main focus today, to our early work on PACAP in the autonomic nervous system, in which it was first shown to be a bona fide neurotransmitter. And I note with gratitude that PACAP was confirmed in the field as a neurotransmitter with acetylcholine at the adrenomedullary synapse in an authoritative review led by Arturo Hernandez Cruz and 3 other confirmed cholinergic biologists. And they graciously allowed me a free hand in the section on PACAP if I agreed to change my name from Lee to Leo to harmonize with the other authors, who were Arturo, Emilio, Ricardo, and Antonio. So just to briefly review, I'm sorry I'm a little distracted because people I'm happy to see keep entering the room. So to briefly review, PACAP is expressed in the cholinergic neurons of the splanchnic nerve that arises in the intermediolateral column of the spinal cord, and its synapses on chromaffin cells in the adrenal gland. Knockout of PACAP in the mouse results in impaired epinephrine secretion in response to prolonged metabolic stress. And that results in the actual death of mice unless they're rescued by infusion of PACAP or by being administered glucose. So PACAP is required for survival from prolonged hypoglycemia, and it's reversible by the addition of PACAP. I'm just, again, so pleased that so many people are joining, even late, but you have to excuse me because each time you hear a chime, I have to let someone into the room. So at any rate, maybe there's a way to kind of close the door, although I hate to do that. At any rate, PACAP is a neurotransmitter in the autonomic nervous system, but it's also expressed in the central nervous system. Does it work with classical transmitters in the CNS as it does with acetylcholine in the periphery? So we've chosen a broad focus, not only on the electrophysiological aspects of neurotransmission in the CNS to do this, but broadly on how synapses function to convey information represented as stimuli from the periphery. I assume that you're seeing my arrow. Stimuli from the periphery to result through a series of neuronal connections called a circuit to behavioral output on the part of the organism. Now note that information flow across these physiological synapses at circuit nodes involves the passage through first messengers, either ionotropic ones like glutamate or metabotropic ones such as PACAP and monoamines like dopamine, serotonin, and norepinephrine. The ionotropic neurotransmission opens ion channels that cause cell depolarization and action potentials. And the metabotropic neurotransmitters cause elevation of cyclic AMP or calcium, but with a time delay that results in activation as well. So a quick reminder about how classical and non-classical transmitters work at the cellular level will be informative as we proceed. First, ionotropic and metabotropic neurotransmitters differ presynaptically because ionotropic transmitters, classical transmitters like acetylcholine, GABA, and glutamate are released from small synaptic vesicles, and they're released at both low and high frequencies. Postsynaptically, the actions of classical transmitters at ion channels are fast, and the actions of metabotropic transmitters postsynaptically are slower because it takes time for cyclic AMP and calcium to accumulate after the receptors are occupied. So the situation at the adrenomedullary synapse really illustrates this. With our colleagues, Corey Smith and his co-workers some time ago, we showed that the adrenal gland could be removed from a mouse, either wild type or lacking PACAP expression. And the splanchnic nerve stimulation can then result in epinephrine secretion at low frequencies of stimulation. I'm sorry, I'm getting a little monitor movement. Anyway, at low frequencies of stimulation, small amounts of catecholamines are secreted when the splanchnic nerve is stimulated. This is now an isolated adrenal gland, so the only PACAP is in these nerve terminals. And at low frequencies of stimulation, it doesn't matter whether PACAP is present in wild-type glands or absent in PACAP knockout glands. But at higher frequencies, like the frequencies you get during stress, there's an increased catecholamine release, and that's completely abolished in the absence of PACAP. So PACAP is solely responsible for the high frequency release at this synapse. So let's move on to the central nervous system. I hope I've reminded you, and if not reminded, convinced you that PACAP is indeed a bona fide neurotransmitter at the splanchnico-adrenomedullary synapse. How does it function in the brain? And we've chosen two brain pathways that are prominent in processing of aversive, stressful, or threatening stimuli by the brain. The first, of course, is the HPA axis, which transduces stress responses into release of CRH to the pituitary, which then releases ACTH into the bloodstream to cause cortisol release into the general circulation. And the second is a very small but critical group of neurons in the brainstem called the parabrachial nucleus that projects to the extended amygdala, including the central amygdala and the bed nucleus of the stria terminalis, and mediates behavioral responses, including protective behavior, avoidance behavior, sickness behavior, etc., to noxious, threatening, and stressful stimuli. So how are these pathways relevant to examining the neurotransmitter co-transmission by ionotropic and metabotropic neurotransmitters, by glutamate, for example, and PACAP? Because PACAP is involved in both of these pathways, and because, as Li Mei Zhang and colleagues have shown, PACAP is co-expressed with the excitatory neurotransmitter glutamate in all of the neurons within this pathway, in the cortex, in the hypothalamus, and in the brainstem, and in the extended amygdala. Now, I mentioned that immediate early genes are invariably upregulated when neurons are activated or brought online during engagement of specific neuronal circuits in the brain in response to external stimuli. A challenge to understanding which type of neurotransmitter is driving long-term engagement, however, is that both metabotropic first messengers, like PACAP, and ionotropic first messengers, like glutamate, can both cause either a cyclic AMP-dependent or a calcium-dependent upregulation of these immediate early genes, like FOS and EGR1. So it's difficult to know which ionotropic or metabotropic signaling is actually driving immediate early gene expression in a given physiological situation. So we begin our effort to correlate PACAP's action across the stress synapse by looking at endocrine and behavioral responses to stress that are associated with differential effects of PACAP deficiency. Restraint stress is the main stressor that we use in our lab, because I'm a pharmacologist, and this is the one stress that can actually be administered in a dose-dependent way. You can either give an hour of restraint stress to an animal, 2 hours or 3 hours, and you can repeat that on the first day all the way through as many days as you wish. And what emerged on applying stress under at different doses is that there's a kind of a dose-related increase in hypophagia 24 hours later. That is, just an hour of stress causes the animal enough anxiety that it doesn't eat for the next 24 hours. And that lack of eating, that anxiety that leads to anorexia, is completely abolished in animals that don't express PACAP. And that's also true if animals are given a chronic stressor. Day one of 1 hour of restraint stress, or 2 hours a day or 3 hours a day for longer periods of time, result in this weight loss, which is virtually completely PACAP dependent. But if we now look at the endocrine effects of restraint stress, we see that an hour of restraint stress, 2 hours or 3 hours, all make cortisol levels go up. But only the 3-hour restraint stress shows a slight PACAP dependence. Now, this is increased, however, if we look at PACAP dependence of chronic stress. And the reason for that is that what PACAP does in the cells that secrete corticotropin-releasing hormone, CRH, is that they elevate the messenger RNA encoding CRH and also increase the immediate early gene FOS. And both of those effects, the FOS increase and the CRH mRNA increase, are completely abolished in PACAP knockout mice. So that's the effect of PACAP at the stress axis. And the examination of these effects of PACAP have led us to a deeper examination of its role in the HPA axis, which turns out to be through an entirely extra hypothalamic pathway that originates in the cerebral cortex and projects down to those CRH neurons. And we hope that this system is going to teach us about some of the unique facets of neuropeptide glutamate co-transmission in the brain. This pathway has both PACAP and glutamate, and we can manipulate the neurotransmitter within this pathway to see what happens during the stress response. And then later, I'll tell you about a second pathway from the brainstem to the amygdala that regulates a separate aspect of restraint stress, which is the hypophagia. So let's turn to the first case. PACAP medial prefrontal cortical PVN projection regulates FOS and CRH mRNA elevation in the PVN after restraint stress. So what Sunny Zhang did was to inject a Cree virus into PACAP-floxed mice into this area of the brain where these PACAPergic neurons reside. And as a result of that, there's a complete, this shows the expression of PACAP in those cortical neurons. It's completely abolished when we lesion with an AAV injection. And what does that lesioning do? When the animals are then restrained, there's no longer an increase in messenger RNA for CFOS, and there's no longer an increase in messenger RNA for CRH. Cord elevation still goes up acutely, but the long-term elevation of cord becomes blunted with time because that CRH mRNA isn't being increased, and the battery, if you will, of the CRH neurons is gradually running down. So we used a combined rabies virus and conditional genetic mouse approach to create mice in which a retrogradely transportable rabies virus would be manufactured only in CRH neurons of the PVN. These are so-called starter neurons here. And then we ask which neurons of the brain actually have a synaptic relationship with these CRH neurons to confirm that this pathway, which affects the function of these neurons, is actually synaptically related to it. And indeed, what we find is that retrogradely transported virus ends up in the PACAPergic neurons of the medial prefrontal cortex. So the PVN is synaptically connected to the same PACAPergic neurons that we had earlier showed support restraint stress-induced FOS elevation and CRH mRNA regulation to maintain long-term stress responses. Now, consistent with this, infusion of PACAP directly into the PVN caused FOS upregulation and CRH upregulation, while glutamate had no effect on either. And this upregulation occurred even in PACAP deficient mice, which supports a direct effect of the exogenous PACAP on PACAP receptors, which is blocked, by the way, by inhibition of cyclic AMP elevation in the CRH neurons during the PACAP infusion, which supports a solely metabolic signaling by PACAP. Just to tell you what this experiment is, we're still infusing a PACAP into the brain, but beforehand, we've instilled into these CRH neurons a phosphodiesterase, which will then block cyclic AMP elevation. So when you infuse PACAP into the brain, you get an increase in FOS and you get an increase in CRH messenger RNA. But when you don't allow cyclic AMP levels to go up, that effect is blunted or indeed completely abolished. So the upregulation of CFOS in PVN neurons after PACAP infusion is blocked by blocking cyclic AMP elevation in these CRH neurons. And the upregulation of CFOS and PVN neurons after restraint stress, like that for direct PACAP infusion, is also blocked by expression of either the PDE shown here or by the protein kinase A inhibitor PKI, which suggests that the effects of endogenous PACAP on stress-induced engagement of PVN-CRH neurons is indeed linked to cyclic AMP elevation. That is, it is metabotropic. Now, CFOS, CRH mRNA in the PVN neurons and cord in blood are also all elevated in PVNC, are all elevated by restraint stress, but they're also elevated by foot shock, indicating that this stressor, like restraint stress, activates these neurons. There's no increase in CRH because the foot shock isn't a prolonged enough stress to cause an elevation in the CRH. It just causes a transient elevation in FOS, which then goes down, and a transient elevation in court levels, which again decreases after 5 minutes of foot shock within several minutes. Now, Baines and colleague previously showed just a couple of years ago that optogenetic silencing of CRH PVN neurons actually eliminates grooming that occurs after foot shock. And activating these neurons, the CRH neurons optogenetically, causes grooming independently of foot shock. So we asked whether this effect on CRH neurons might be mediated by the frontocortical PACAP pathway. And we asked that by looking at the grooming behavior when PACAP is knocked out in the hypothalamus. PACAP producing cells of the hypothalamus are no longer making PACAP, no effect on grooming. But if we knock out PACAP just in frontocortical neurons, there is an abolition of this shock and foot shock induced grooming. And this PACAP dependence of foot shock induced grooming is due directly to PACAP release from this mediofrontocortical pathway, because if we knock out PACAP just in that pathway, we also knock out the grooming behavior and also knock out FOS induction. Now, to directly test the role of cyclic AMP elevation in stress responding, that we know is initiated by PACAP release and that is cyclic AMP dependent. We took advantage of a reagent that was developed by Andrew Ludis, a colleague of ours here on the NIH campus. Andrew and his colleagues have developed a blue light activated adenylate cyclase. When this adenylate cyclase is expressed in neurons and an optical fiber is introduced into the brain to shine blue light on these proteins, on this protein expressed within those cells, cyclic AMP is elevated. So we wanted to know, would this independently cause grooming if BPAC was expressed in the CRH neurons, and then we used optogenetic stimulation of them? And in fact, with blue light stimulation, you can see that with the onset of blue light stimulation, the animal immediately begins a bout of grooming. And that bout of grooming continues until blue light is turned off. And then the animal goes back to its usual activities. So a 5-minute stimulation with blue light elicited grooming after an initial delay of a little bit under a minute, and continued for the duration of the light exposure and stopped after light exposure stopped. So this is going to, there we go. So this is going to allow us to ask, what are the temporal requirements for cyclic AMP elevation for mimicking the effects of foot shock during a 5-minute period on subsequent grooming behavior and FOS elevation or CRH mRNA elevation over the subsequent 30 minutes. So we set up this experiment to administer, just like I showed you, for 5 minutes, blue light for 15 or 30 minutes, and then at the end of the 30-minute period, collect brains for in situ hybridization histochemistry, tail bleeding for cord elevation, and let's see what the result of cyclic AMP elevation is. Now, what happens optogenetically is that there's a quite closely coincident with the duration of elevation of adenylate cyclase and cyclic AMP elevation, onset and duration of grooming caused by 5 minutes or 15 minutes or 30 minutes of cyclic AMP elevation. So turns out that 30 minutes of cyclic AMP elevation looks a lot like the results of 5 minutes of foot shock. If we elevate cyclic AMP in these neurons for 15 minutes and then look 15 minutes later, there's no increase in FOS. But if we elevate cyclic AMP continuously for 30 minutes, FOS is up by 30 minutes, and no effect on CRH at this time, and no effect on cord elevation. So the cord part of the stress response is really completely independent of cyclic AMP. So now we've linked cyclic AMP or metabotropic neurotransmission to some FOS elevation, CRH biosynthesis and grooming, but not all. The release of CRH itself that leads to cord elevation is mediated through cyclic AMP. CRH release, presumably by some ionotropic neurotransmitter not yet identified. It's unlikely to be glutamate, but we are still looking for the second neurotransmitter that exerts this parallel effect. So the picture that we're getting is not that neurons are simply activated in a circuit in some all or none way, but that certain aspects of neuronal engagement are activated metabotropically, and other aspects of neuronal engagement are mediated ionotropically. Let's take a look at the other stress circuit that I mentioned. Hypophagia and FOS induction. FOS induction occurs in this pathway after restraint stress, and the hypophagia that results after restraint stress also depends on the presence of PACAP in this pathway. So these are two separate experiments where we've knocked out, these are the PACAP terminals in the central amygdala. They're gone if we knock out PACAP in this set of brainstem neurons. Likewise gone if we knock it out in a separate experiment, instead showing it in a different color in this case. And what we find is that FOS induction in these target neurons in the amygdala no longer goes up after restraint stress. And weight loss is severely blunted after restraint stress. And in fact, if we silence these neurons chemogenetically, the neurons that are the targets of the PACAPergic projections, we also blunt the weight loss caused by restraint stress. And this synapse is particularly important as an issue of co-transmission because it contains both PACAP presynaptically and glutamate-associated proteins like GLUT1 and the NMDA receptor postsynaptically, as investigated by Li Mei Zhang's lab using focused ion beam scanning electron microscopy to look in detail at this synapse. The synapse appears to constitute a specialized synaptic structure that enwraps the target neurons in a kind of cup or calyx, similar to the calyx of Held glutamatergic synapse of the auditory pathway in the brainstem, which may account for its unique co-transmitter properties, as I'll show you now. Vito Hernandez, on a visit to the lab, examined the electrophysiological effects of PACAP on these PKC delta expressing target neurons in the central amygdala. And not only does PACAP application appear to increase the excitability of these neurons in slice preparations, but it does so through a mechanism that's apparently related to a long-term change in resting membrane potential that biases them towards excitation at lower input thresholds, which may be relevant to the differential effects of glutamate co-transmission for stress and pain processing by this circuit. And I'm going to finish up with that now. Before I do that, I want to point out, and this gives me a chance to feature some experiments that Diego did when he was here at the NIH. He expressed channel rhodopsin and PACAPergic projections to the central amygdala, and then in slice preparations, used these neurons, now armed for optogenetic activation, to show that blue light shining on these terminals releases a transmitter whose excitatory effects can be measured in the postsynaptic PKC delta neurons, and those signals are abolished in the presence of CNQX. So this is glutamatergic transmission that's occurring in this PACAPergic pathway. And yet, when we knock out VGLU2, so that glutamatergic transmission is no longer occurring in this pathway, there's no effect on either the body weight loss after restraint stress or on FOS induction after restraint stress. Both of these are PACAP-dependent phenomena. Neither of these are glutamate-dependent phenomena. Now, we've identified in collaboration with the laboratory of Jaromir Carrasquillo at the NIH another function for PACAP in this pathway, and that's to mediate pain sensitization after nerve injury. So mice with a compression of the sciatic nerve show an enhanced aversive response to the application of acetone, to tactile stimuli. This is increased pain perception going in this direction. So that's increased by sciatic nerve damage in otherwise wild-type animals. This is increase in pain perception in response to tactile stimulus, pressure, or heat. And that effect is greatly increased in wild-type mice with sciatic nerve injury. But in PACAP knockout, constitutive knockout mice, that effect is of cold stress, of tactile and pressure stimulation, and of heat is completely absent. Allodynia, or increased pain sensitization, requires PACAP. Does it require PACAP in this particular pathway? Yes, it does. We get the same results on abolition of the heat response and the pressure response that are seen in wild-type mice if we knock out PACAP from this pathway. And if we knock out VGLU2 from this pathway, we also see that the injury-induced pain sensitization is abolished. It's abolished in the animals that no longer express VGLU2 in the pathway, still present in control animals in which we don't abolish VGLU2 expression in the pathway. So as the lab of Sung Han has recently showed, glutamate neurotransmitter neuropeptides are released mutually exclusively at low and high frequencies of stimulation, respectively, in this pathway. So our results here suggest, if that's true, that restraint stress responses through this circuit reflect mainly high-frequency stimulation, where PACAP is released preferentially, while pain sensitization may require alternating high and low frequency, releasing both glutamate and PACAP during the development of pain sensitization and allodynia. So I want to close by noting that we feel we're starting to link cyclic AMP elevation in two separate stress circuits to specific aspects of signaling, exemplified by FOS induction in both cases, both in the CRH neurons of the hypothalamus and the PKC delta neurons of the central amygdala. But also there are downstream behavioral consequences, some of which are affected by glutamate in the case of the PKC delta neurons, and some of which are not affected or driven by glutamate in the case of CRH secretion and cord elevation in the case of the CRH neurons. So is there a separate role for glutamate and and peptides? Is co-transmission necessary in every case? Often, but not always. I've given you examples where PACAP and glutamate are both required for the overall function of a synapse in pain sensitization, and where only PACAP is required for the hypophagic effects of restraint stress, and then another system in which PACAP mediates both immediate behavioral responses to foot shock and also long-term responses to chronic restraint stress, but doesn't seem to be important in the CRH release that immediately occurs upon the application of stressful stimuli. So with that, I want to thank the members of the lab, Sunny Zhang, Wen Shu, Haiying Zhan, post-bac student William Brancaleone, and Sam Hargrove, also a post-bac in the lab, as well as those that have preceded them. And of course, also the work couldn't have been accomplished without our colleagues in the IRP, Chip Gerfin, Ugo Tejeda, with the help of Hector Yarur, Ted Usden, and Sarah Avram in our imaging facility, Yogita Chudasama in our behavioral core. It's been a pleasure to share this work with Limei Zhang and Vito Hernandez and with Diego. And also we thank our colleagues Yarimar Carrasquillo and her associates, Sudaman Singh and William Fortun Rosada, and Andrew Ludis, who has guided us through the use of these really important cyclic AMP-dependent in vivo deployable reagents to get a new focus on exactly how metabotropic signaling works together with ionotropic signaling in the brain to engage and fulfill stress circuits. Thanks.
Thank you, Lee. I'm not sure if you can hear the clapping. So thank you, Lee. Always is a pleasure to listen to you. So let's move to the question if there's any. ¿Tiene alguna pregunta? ¿Fueron siguiendo ahí los subtítulos? ¿Hola? Sí. Yo también tengo una.
Very, very. Baru Akin. Highly beautiful talk. Congratulations.
Thank you.
I just would like you to make a little bit extensive explanation how this PACAP can activate the immediate early gene such like force. Because I think this is a
Limei, your words aren't quite coming through to me. I'm seeing a few of them on the caption, but only a few. Could you repeat what you just said?
I was asking if you can make a little bit more explanation about the role of PACAP through the GPCR the G protein coupled receptor to activate force the immediate early gene this pathway. If you can explain a little bit more.
Well, thanks. I'm happy to do that. What we do know about this pathway, and we know the most about it, of course, in the CRH neurons of the PVN, is that the upregulation of FOS and the upregulation and the increased transcription of the CRH gene are both blocked after restraint stress if we introduce the protein kinase A inhibitor pKa, or if we block the elevation of cyclic AMP with a phosphodiesterase. And so that implies the most straightforward explanation is that the PAC1 receptor is engaging the GS G protein, and that is activating adenylate cyclase. The activation of adenylate cyclase elevates cyclic AMP, and cyclic AMP, of course, activates protein kinase A. So we predict that this occurs through the phosphorylation and activation of the protein CREB, and it's CREB that interacts with the FOS gene. FOS itself is a transcription factor, and it appears, at least by the mutual effects of blockade of FOS induction and then later of CRH induction, that the CRH gene transcription is probably FOS dependent.
Thank you, Lee. I would like you to clarify the PACAP, the endogenous receptor PAC1. Is this PAC1 only the GQ receptor or also GS receptor? And if it is both, how you can explain the effect that you have observed?
Well, there are signaling pathways. I mentioned the signaling pathway through GS, where cyclic AMP is directly increased because adenylate cyclase is activated. We haven't ruled out that PACAP could be acting through GQ. GQ, by activating phospholipase and increasing inositol trisphosphate, causes an increase in intracellular calcium. There are, in most neuronal cells, calcium-dependent adenylate cyclases. So it could be, and it would be consistent with all of the data we have so far, that PACAP is either activating GQ, which leads to a calcium-dependent adenylate cyclase activation, or PACAP is activating GS, which is causing a direct increase in adenylate cyclase and cyclic AMP elevation. That's going to require looking at the effects of knocking out these G proteins one by one in the CRH neurons. And we have tools to do that, and we're going to try to do that because that's probably an important avenue to pursue.
Thank you very much.
Thank you, Lee. I have a question here from Zoom. So, what does the significance of the neuropeptide transmission for the studies on the cutting edge of the pain and aversion modulation? I mean, which could be the differences with the classic neurotransmitters?
Well, there's a difference between potential therapeutic action of or modulation of classical transmitters, but there's also the larger case of avoiding opiates. So there are two cases in which opiates are not good analgesics. And these two phenomena are thought to contribute to the opiate epidemic among people who are not classically subject to opiate abuse, but they become opiate addicted through the use of opiates as medications. And those two things are opiate-induced hyperalgesia, in which long-term treatment with opiates actually leads to a hyperalgesia of the type that I've described in these animal models. And in addition, there are chronic hyperalgesics, chronic pain states in which people repeatedly take treatments, and that leads to potentially to opiate addiction. So one of the things we're doing with our colleagues at the National Center for Accelerating Translational Medicine is looking to see if we can find peptide antagonists of the PAC1 receptor, which might be used for the treatment of either opiate-induced hyperalgesia or just chronic inflammatory allodynia pain sensitization, because people that suffer from these hypersensitive states, it really is a kind of a lifestyle changing clinical condition. People complain that it's even painful to wear their clothes throughout the day because they have a generalized pain sensation throughout the skin as an organ. So this is an area where there might be a way to shift to a new therapeutic target.
That's good. Thank you, Lee. Finally, just take advance here because Lee, here in the auditorium, we have young people interested in neuroscience applied to veterinary medicine. What your thoughts about from your experience through 4 decades studying the nervous system? You could give us today for them about the neuropeptides in veterinary research. So you have some thoughts about this.
Well, I'm gonna save some of those thoughts, Diego, because I plan to visit again during Brain Awareness Week,
Sure.
and I hope to give a less technical, more general talk on that very subject. But just to say briefly, animal models are critically important for preclinical testing of the likelihood that something is going to be a drug avenue. So, for example, in migraine, PACAP antagonism, as well as CGRP antagonism, are being developed clinically. But the best way to do that depends on it working properly in an animal model. And finding the correct animal model is also of key importance. There's an article in the literature that says that we've found 1,067 treatments for stroke damage in rodents, but not one for stroke damage in humans. So if we're trying to avoid the clinical problems associated with stroke in rodents, we're doing great. If we're trying to avoid it in humans, not so much. And a lot of people attribute that to our incorrect use of animal models. Many people think that the right way to do this is to jump from the mouse and the rat into the clinic. But more thoughtful therapeutics developers realize that there are as many animal models as there are types of animals. And it's very important to know the comparative endocrinology, the comparative anatomy, the comparative physiology and pharmacology of different mammalian and even non-mammalian animal models, especially as we're starting to see so many promising treatments now for Alzheimer's disease. We went decades with no treatments for Alzheimer's disease except to inhibit acetylcholinesterase. And now there seems to be dozens of potential treatments. We even have too many because it's not possible to test all of these in patients. So we need to start with mouse and rat models for basic feasibility, but we also need to rapidly move with the most promising drugs to more complicated animal models that better mimic the human condition.
Thank you, Luis, so much because it's really interesting. And, all the time, my NIH studying, I was interested to apply this neuropeptides transmission to veterinary research. So that was, I think, is a very good opportunity to talk about this today. We have another question here. So, Lee, how could PACAP? Okay, can you hear me?
I can hear you.
Oh, thank you. How could PACAP affect the circadian clock response to blue light?
I actually was going to briefly mention something about circadian rhythms. My colleague in the NIMH, Dr. Harar, has found that one can crash the gate for phase shifting during the daytime by manipulating the retinohypothalamic pathway, and some of its specific neurotransmission involves a PACAP. We've found that in PACAP knockout mice and in the PACAP knockout SCN, that in fact PACAP is really important in phase shifting. So lack of PACAP in the retinohypothalamic tract really doesn't have any effect on phase delay, but phase advance, which is totally dependent on glutamate, whereas phase advance is dependent on both glutamate and PACAP being present at that synapse. And we're still not sure why that is, but it's another example, like the synapses that I showed for stress, that where classical transmitters interact with neuropeptide metabotropic transmitters, but in very, very specific ways.
Thank you, Lee. Have a question here. Another question. Do you think PACAP antagonists could be effective, or would they need to given early to prevent chronicity of pain?
Well, that's a really great question. I alluded to Samar Hattar's work on circadian rhythms, and he thinks that that's quite applicable to treatment of jet lag. But if in that case, one would have to be very careful about using either a PACAP agonist or a PACAP antagonist. Presumably, they would shift circadian rhythms in opposite ways. We know that PACAP is has a very positive effect on retarding atherosclerosis in animals fed a high-fat diet, because blocking PACAP's action accelerates atherosclerosis. We know that for pain sensitization, we would want to give a PACAP antagonist chronically. Would that have an effect on PACAP's effect to help block atherosclerosis? And part of the key for that, I think, is going to come to developing drugs that are specific for delivery to the brain, where the effects of PACAP in pain sensitization would be felt, and in the periphery, where PACAP's effects on atherosclerosis and on migraine would be felt. So a lot of refinement is going to be necessary. But the original question, I'm sorry I strayed from it, was would the treatment with a PACAP antagonist need to be chronic in order to avoid allodynia? And I think that's a very interesting question, and one that's very difficult to fully answer, because pain sensitization is a chronic state. But what events lead to the development of that chronic state? If people develop chronic pain sensitization after an initial injury, then maybe treatment with a PACAP antagonist just around the time of injury would permanently ameliorate or delay pain sensitization, and then no further treatment would be needed. So we really need to work out the dynamics of these effects before we can answer that question.
Thank you so much, Lee. All right, so I think we're done with the questions. So again, Lee, congrats. Thank you so much for this interesting talk today. And let me just to say bye here to the auditorium. Bueno, los esperamos el siguiente mes, el siguiente seminario de los tópicos, que será el próximo mes. Pues estén al pendiente ahí con la Secretaría de Educación Continua, y también les agradecemos allá a los chicos de cabina por hacer esto posible. Pues muchas gracias, chicas, chicos, y pues ojalá les haya gustado la plática. Hasta luego. Thank you, Lee. Just wait a moment there, Lee.
Yeah, I'll stick with
Oh,
you.
yes, yes,
Let's
yes, could you
pretend
please? Let's pretend exactly.
let's pretend I'm actually there, standing at the podium, and we're just hanging around for a bit. So if anybody wants to come up and ask any other questions, I'll stay around for a few minutes, Diego. And
Thank you.
unless you have other places to go, then I'll say goodbye.
All right, thank you so much, Lee.
I see you're standing with Limei.
Right here.
I think I can turn off or I already turned off the share. Yeah. Okay, good.
Yes, I'm here.
Lee, can you hear me?
I sure can.
Lee, I just would like to extend an invitation for the next semester for the graduate school that we have been organizing this course. If you can accept from the second week of September, the last week of October, Aaron Anansaran just said he is very happy to accept my invitation. So I would like to organize that both of you to come more or less the same time. So we will have a week of intensive class. So we can
That'd be
take
awesome.
advantage. Yeah, if you can check off your calendar,
Yeah.
the second week of September, because first week of September, we are in Chile. You remember we have a commitment in Chile.
Yep.
Yeah, so we will come back this first week, September, from Chile to our lab. So from second week to the last week of October, you choose 1 week or 2 options. So I can tell Aaron, because he will be teaching, but he said if you can decide, he will help. He will find someone to substitute, to replace him.
At the very moment, my calendar is open. The only days I have to worry about are Grandparents' Day on the 13th and Yom Kippur on the 20th.
Don't say that, Lee. That's not very correct. Yeah, so come in, I think the first week, second week of October, I will tell Aaron so he can arrange the teaching. And the second thing, I need you to send me a acceptation letter. So I'm going to prepare the invitation letter because the graduate